My name is Ross Kagan. If you haven't figured that out by now, I'm a precision Regis Professor of Precision Medicine at University of Glasgow. I'm a fly boy from way back. We're going to talk about flies in a bit. My lab's mostly developing cancer therapeutics. Some of you got to see a little bit of the technology yesterday with Laura's talk. This is the third and final day of this pretty amazing Drosophila conference that I'm actually pretty excited about. On day one, we heard from Adam Dobson on how bacteria and diet sort of work hand in hand to extend lifespan. We heard from Amos on how flies can be used to study toxicology. From Nicole Broderick on how the fly microbiota affects development. Carlos Rivera about how you can show insanely cool pictures, but also how the fly feeding behavior is a great paradigm for studying behavior. On day two, we heard from Hamid about migrating cells, so-called border cells, and how they migrate in the Drosophila egg, and also an overview generally of how you can use Drosophila and Drosophila embryo to study key questions in development and disease. We heard from Tano Gonzalez, a leader in using flies for cancer, on his 9 questions in cancer. We heard from Laura Tauert, who is founder and CEO of My Personal Therapeutics, which is a UK company that uses flies to develop treatments for cancer. And finally, we heard from Linda Partridge, who basically discussed some of their work on developing treatments, if you will, for longevity, or at least health longevity, improved health for diseases based on aging. So a few words, where do we go from here? I know there's both excitement, but there's also trepidation, or, how do we push forward? We have a pandemic, there's lots of roadblocks moving forward. This, of course, is up to all of you. We have just brought to you some speakers that we know are good and we thought you would find valuable. We're hoping, and I think it's seeing the questions over the last 2 days and all the participants. My personal wish, and I'm going to talk about me, my motivation is I'm a big fan of science in general and Drosophila in particular and its role in it. I'll talk more about that in a moment. And we're always going to need the newest, brightest young minds to keep this field strong. And I think a lot of those clearly are in Africa. And so that's my motivation, is to pass the baton that was given to me to the next generation. And many of you are it. I want to give a shout out to the DrosAfrica family. The founders, Lola Martin Bermudo, Isa Palacios, and Marta Vicente Crespo. But also this was put on as a real team effort. Jose Casal, Hayat Salami, Wassam Meshrif, and Luca Gabel have really done important work, as has Sophia. I didn't list her here, who has gotten, has been keeping an eye on Slack. And yes, Slack has really been going. And I know that what really makes me excited is that there are already more and more efforts, in addition to DrosAfrica, in Africa, to drive this model system forward. Hamid gave a shout out yesterday to Droso for Nigeria from Rashidat Abdulaziz. I also want to point out, she was kind enough to contact me actually multiple times over the last many months. Helena Chukwu Kwanashi and her Zaria Droso Club, also coming out of Nigeria. And I hope that these efforts and other ones are started. As my mother likes to say all the time, so she's really angry about what's going on politically in the US, she said, you can complain about things or you can do something about it. And she has personally made it her goal to beat Trump by herself. And frankly, she's winning right now. So she's doing all right. So like others, please share your questions in the chat room and in the chat here. And then either I'll read it, or as you saw, if you just say, hey, I have a question, allow you, we'll put you on to ask that question. I think maybe that got lost in the shuffle, but please feel free to do that, or I'm happy to ask those questions as well. So this is really a lot of fun for me, so let's keep building this structure. Some housekeeping stuff about Zoom. Group picture. So we're going to have 2 talks today as part of our development session from Aissette and from Julia Cordero. And then we're going to have a 15-minute break, mercifully. But a few minutes of that break, at the very beginning of it, we're going to do a group picture. So that means please turn on your camera, and then we will take snapshots across it. Actually, several people from the DrosAfrica group will do that. So we'll make sure we actually capture them. So please turn it on. And if you were thinking of just bailing for 15 minutes and not doing that, why would you miss out on this opportunity to get yourself, put your beautiful face put up in lights? So hang out a little bit, put your camera on, good to go. So let's see what else. As I just did, if you're trying to figure out how you keep getting muted, that's me. And that's just to keep the general chaos down. But if you have a question to ask, please do so, and you can unmute yourself, and it's all good to go. I do have one last poll, I hope. Yeah. And this question is, how did you hear of the DrosAfrica Online Workshop 2020? So I'm launching this poll. I've given you 3 choices, including the ever popular other. And we'd like to know how you knew about this. There have been so many people that have signed up for this. It's really helpful to know. So we'll give about a minute. Ah, cool. I love polls. Meanwhile, let me switch my background to here. All right, groovy. I think we have the majority. So I am going to end this poll. And as you can see, or I'm about to show you, so pretty evenly split between social media and sharing with friends. So now that we have so many people that have been part of this workshop, this is great. So you can continue to spread the word, please do. The beautiful thing about doing science is that it is more than almost anything, really a community of folks. So I love that. Okay, cool. So my tradition to talk about flying Nobel Prizes and some of the history and background behind it. My third one, I'm going to end with the most recent Nobel Prize from a couple of years ago, 2017, circadian rhythms. I'm going to tell you a story that is probably true. I've been told it by people who should know, but I was told many years ago and then I haven't heard it since, but we're going to go with true because they have no reason to make it up. And that is that, as I mentioned, Drosophila, which was really the founding model for understanding developmental genetics, it really kind of went, I wouldn't say off, but quiet for a while. And that really reflected also developmental biology went quiet for a while. In fact, developmental biology used to be called embryology. And then they, just like genetics became genomics, they changed embryology to developmental biology because it just sounded cooler. That's why old papers refer to embryology and new papers refer to developmental biology. And journals change their names and all that stuff to just try to get on the cool chemical biology, you know, et cetera, naming. And after World War II, there was a group of physicists, one of them Seymour Benzer, another one Sidney Brenner, another one Gunther Stent. These were physicists that were challenged by some of the preeminent physicists. Basically, the idea was that physics had had its day. It had figured out all the difficult problems. Quantum mechanics was worked out by that point, much of it anyway. Relativity was already a thing. And physics seemed like it wasn't the sort of happening thing anymore. So what was going to be the happening thing? Biology. And there was actually a famous book written basically challenging physicists to take up the baton of biology and solve some difficult problems, like especially neurobiology. And there's a group of these physicists who really got together in the early 1960s, and I'm told literally, physically got in a room and had a conversation about how they were going to pursue what we now call developmental neurobiology or neurobiology. And so Sidney Brenner said, I'm going to work on this little roundworm that we found because it's very simple. I'm going to stent. I'm going to work on aplesia because its nervous system is pretty simple. And Seymour Benzer, who had actually arguably invented the concept of the physical gene by his work in R2 viruses, he said, no, I'm going to go and work on Drosophila, fruit flies. And because they have genetics, and I think I can work out some interesting things. And so he actually began to find some of the first mutations that affected behavior. He found a series of these. And in fact, when Seymour was working, we were talking about into the 70s, into the 80s, really, what he was doing was very controversial. People said, oh, you're going to work out the human consciousness in the brain using genetics? That offends me. I don't like that. And one person in particular, well, remain nameless, was a Harvard professor, was especially offended and circulated a letter saying, what is this crap? And Seymour didn't really care, but his friends told him, you really need to defend this approach because otherwise it's going to become a problem. And in those days, there was scientific community was very small. Everybody knew each other. So Seymour gave what apparently was a very famous talk where he suddenly revealed all these mutations in circadian rhythms, in choice tests where flies would run towards light, negative geotaxis, a lot of these behavioral mutations that affected that. And he gave this talk and he laid them out. And so this person who had circulated the letter then stood up and said, so basically you're saying you're going to find the gene for consciousness. And Seymour famously shot back, well, if you define consciousness, we'll do a screen. Why don't you give me a definition? And that's when this person realized that they actually had no idea what they were talking about when they said consciousness, that it was just some vague word. And the person shut up, they sat down, and that was considered the birth of neuro or behavioral genetics, neurogenetics. And Seymour had spun out many different disciplines. And one of them he spun out was circadian rhythms. Ron Konopka in his lab found the first period mutants that, the first mutations that affected circadian rhythms. And the basics of circadian rhythm was worked out by these three gentlemen, Jeff Hall, who worked through the genetics of it, and Mike Rosbash and Mike Young, who cloned the genes and showed biochemically how they worked, of the whole timeless per axis that works within each cell in our body to set a clock. And this is why if you're woken up at 3 in the morning, you feel weird. You feel it's difficult because your body has an intrinsic clock. They got the Nobel for that. And each of them in their own way, especially Jeff, is really quite a character. I've known all three of them for many years. So that's my Nobel story. Again, just trying to inspire you because I expect the next Nobel to come through from one of you listeners. Okay, so let's get to it. Today's talk, right on time. Today, we're gonna work on development, which has classically been the strength of Drosophila. Therefore, it has been especially important that we bring in really some of the most powerful people in the field. So we have Aissette Baanannou from Tunisia, who's going to talk about cis regulatory programs. We have Julia Cordero, a friend and colleague of mine here in Glasgow, who's going to talk about the adult intestine. I can't speak today. Then we're going to take a break and have our group picture, so don't forget. Then Eugenia Pedini will talk about cell competition. Monica Betancourt-Diaz will talk about cytoskeleton in development. And then we're going to have a special treat. We're going to take another little break. And then we're going to have Mahmoud Abdel Adi from Egypt is going to talk about career advice. So for those of you who will find that useful, please stay tuned. This is really an incredible opportunity. And also Mohammed Salam, some of us know, we're hoping to get him yesterday, but the good news is he was willing to give us a talk today. And so he's going to really wrap this conference up with discussing the role in the Center of Excellence in Research. So that's a lot. We need to get going on this. And so Aissette, I'm going to hand the baton to you. Please share the screen and let's do this.
Okay. So I start. So hello, everybody. I'm very happy today to speak and to participate to the DrosAfrica workshop. Today I will speak about Drosophila appendage development and my previous work in the group of Henri-Marc Bourbon in Toulouse that lead to the identification of cis-regulatory BAB genes in leg and antenna. So let's start by giving some what's the cis-regulatory model. Donc, cis-regulatory model are a sequence, DNA sequence that are usually about 100 pair base and that are in length and that are situated it can regulate the expression of target genes. So, now this is the cis-regulatory model that will interact with the co-activator complex and distal transcription factor to activate the transcription, the gene by interaction with transcription initiation complex. Donc, there is three types of cis-regulatory model. Enhancers, which regulate gene expression positively, silencer that turn off expression of genes, and ancillary which work indirectly by interacting with other nearby cis-regulatory model. And as you show here, this is an enhancer, but in the presence of ancillary, this ancillary will block the interaction with the promoter, and only this enhancer will activate only this promoter. Current model of enhancer are enhancosomes that in this situation, all the transcription factor must bind to the enhancer and to be active. And there is a second model, billboard, which in this context, only few transcription factors are necessary to be active for the enhancer to be active. Depending on the cell and the time, the cell will choose which factor must be bind to the enhancer. And the transcription factor collective, in this context, there is a set of transcription factors that can bind to different enhancer, and this transcription factor have the ability to interact, which mean that it's not necessary to have all the binding site for this complex to be active. There is generally the enhancer are there is a minimum 2 enhancer for one genes. One there and this which we call redundancy. If we delete one, we will lose the expression of we will not lose the expression of the gene. However, in a certain condition, for example, high temperature, we need the presence of the two CRMs to have a normal expression. Otherwise, we will have a phenotype. Sometimes it can lead to lethality. And this enhancer can interact cooperatively to activate the target genes, or dynamic alternative activation. And each time one enhancer will activate the target gene. Experimentally, this regulatory model can be identified through chromatin immunoprecipitation directed against the transcription factor or histone post-transcriptional modification. There is a new techniques now for identification of enhancer like ATAC-seq assay for transposase accessibility chromatin, and the fair formaldehyde assisted isolation of regulatory element. But the gold standard test to identify the cis regulatory model is to clone the cis regulatory model upstream a promoter which drive the expression of a repertoire gene like Z, GFP, RFP, or luciferase, and then we analyze the expression directly in the embryo or the cells. De novo, emergence of transcription enhancer and modification contribute largely to innovation within genetic program, which lead to help the species to adapt to the environment and to generate new fates that are important for adaptation to the ecological niche. Like arthropods, there is a great variation in number, shape, and function of appendage, which allow multiple purpose from locomotion to feeding or environment sensing. Donc, the Drosophila have the capacity, is holometabole. It go through a complete metamorphosis, which it's include 4 stages.
Which one are you doing first?
What? Which include egg, larva, and the pupal stage, and finally the adult stage. During this metamorphosis, the larva will develop its appendage from imaginal disc, which are epithelial sac-like structure in the embryo that will grow and pattern during larva development. And during the pupal stage, this imaginal disc will evert, like here, the imaginal leg, imaginal disc, will evert and differentiate 2 legs in the adult. This group at stage 10 of embryogenesis, a group of cells, about 30 cells, will start to express distal less in each thoracic hemisegment, and then with the input from other morphogen, DPP and wingless, and the repression induced by EGFP signaling, it will some cells will migrate to the dorsal part of the embryo, and this group of cells will express some genes that are specific to the dorsal primordium, and this dorsal primordium will give rise to the wings or the halter, whereas this primordium, ventral primordium, will give rise to the leg, and with the 6 imaginal disc, we will have six leg in the insects. So these genes are also controlled. This subset of cells are also controlled by the expression of homeoHOX gene, and this is an example when we lose the expression or we have extension of the expression of ultrabithorax. There is a mutation that generates the appearance of a second pair of wings in Drosophila. So after in the leg at the third instar larva stage, the leg disc will express ingril in the dorsal in the posterior region. This will lead to the expression of hedgehog with that will induce the expression of two morphogen, two regions that will produce morphogen. This morphogen will induce the expression of distal S in the center of the disc and repress other genes like DAC in the middle and in the same time and induce DAC in the medial part of the leg. And finally, they will also induce the expression of vein, which will activate the EGFR pathway, and all this will lead to the appearance of many domains in the leg. Finally, with the Drosophila leg is will develop 10 segments which are articulated to each other by a characteristic joint, and in this part, the tarsus we will have will be divided in 5 segments. During my PhD, I have studied the expression, and I analyzed and I identified the cis regulatory model implicated in the regulation of 2 paralogous genes, BAB1 and BAB2. Donc, these genes are important for leg segmentation. And as you see here in wild type, there is the tarsus will be divided in 5 segments, whereas with the mutant BAB1, BAB minus, we will lose the junction and the region is shortened. The same thing will happen in the antenna, and for BAB2 there is a predominant role because BAB1 mutant don't give any phenotype, whereas BAB2 mutant give a phenotype. So the expression of these 2 genes is dynamic. It starts in disc shape in the early L3, and then it will be defined in 4th ring in the late L3 and 2 ring in the antenna. And there is it was many transcription factor and pathway signaling was described as activator and inhibitor for BAB2. The EGFR pathway was described as inhibitor, and BOL was in the distal part, and BOL was described as inhibitor in proximal and distal part, whereas distal S was described as activator, also spineless. However, no evidence of a direct interaction of this transcription factor was described before. So, starting from a systematic genomic work for BAB enhancer that was done by the group of Williams, they have identified 2 regions of two regions that span almost 11 kb that express GFP in leg. We have analyzed after dissection, the expression of this leg, GF, the expression of BAB, and compare it to the GFP, and we found that the same expression, the same expression for the element, for the two element, and the BAB2G. So we did many construction. We deleted many region, and finally we found that the overlapping region is sufficient to have to reproduce the BAB2 expression profile. So we analyzed it by alignment of the Drosophila sequence available in the database, and we found a region, very conserved region that span almost 500 per base. Donc, we have checked that this region that we call it leg antenna element is sufficient to express, to reproduce the pattern of BAB2 in leg and antenna. Then we have tried to check if the equivalent region from virilis, that is far from this melanogaster, if this region is enough to reproduce the pattern of BAB2 in Drosophila. So we cloned this, we generated a transgenic line that expressed GFP in under the control of the leg antenna element of Drosophila virilis, and we found that this element was also sufficient to reproduce the pattern of BAB2 in leg and antenna. Then we have asked the question: is it this element, leg and antenna element, sufficient to reproduce, to save the mutant, to rescue the mutant BAB? So what we did, we constructed, we have integrated a back which contain all the gene BAB2 and the intergenic between BAB1 and BAB2 with the leg antenna element, and also we generated a second back without deleted from this region, and we integrated these two constructions in Drosophila in a context where BAB is mutant. And what we found, that when that this back is enough to rescue the phenotype, and we have something that is like the wild type. However, if we delete this region, we will lose the expression, we will lose the rescue. So this experiment confirmed that this element is necessary for, it's sufficient, it's necessary for BAB2 expression. We have analyzed also the expression by using antibody, and we found that the deletion of this element is induce totally the loss of expression in leg and antenna. Subsequently, we have analyzed, we have tested the capacity of this leg antenna element to rescue the BAB minus phenotype. So so we have cloned this element with the BAB2 cDNA, and we generated a transgenic lines in context of BAB minus mutant. And as you see here, we have rescued the phenotype using only this element, which prove that this element is necessary and sufficient for BAB2 function. Subsequently, we have tried to identify which factors are implicated, which transactivator are implicated in the leg and antenna element activity. So we deleted from sequence from the 3 primes. And as you see here, when we delete some region, the intensity of the signal decrease. Same thing for the antenna and the leg. And if we delete the box, the conserved box too, we have also loss of expression in the distal part of the imaginal disc. The same thing was observed for antenna and for leg. We did also deletion. We tested the effect of deletion from 5' region, and as you see here, when we deleted only 33 per base, we have lost the second ring in the antenna and the 2 ring in the proximal ring in the leg. However, when we delete also the CR1, we lose all the expression in totally the expression of the transgenic line. Finally, we tested if the CR1 is sufficient for to express to drive the expression of GFP like BAB1 and BAB2, but we found that it's not sufficient. We substituted some sequence from this region, this conserved region that we found that is important for the activity of the LA element. So, and we found that some regions are more important than others. For example, this one, LS2, the mutation of this region lead to totally loss of expression. Almost the expression of almost all the activity of the transgene. However, when we mutated this region, we found derepression. So this region could be implicated in the control of inter-ring repression of BAP2. And we proposed this model where only a leg antenna element of about 500 per base is constituted of regions that are important for specificity and second region which is more important for intensity. And we identified in this cis-regulatory CR1 a region that is essential for activation and another that is essential for inter-ring repression. So the second step was to identify which factor, transcription factor, bind directly to this identified region. So we generated the distal-less clone, and we tested before this. We tested if the leg antenna element was controlled in the same way as the endogenous BAB gene. So because distal-less is an activator of BAB gene, we generated a clone for distal-less, and what we found that this clone we lose the expression of BAB2 and also the activity of our transgene, of our enhancer. So we confirmed that this element will have the same regulation that the endogenous genes. We analyzed the sequence of the site of the CR1 box, and we found that we found the presence of 2 sites that can be a consensus site for distal-less. We mutated these 2 regions, and we found that we have tested by EMSA if these 2 regions can bind the protein, And we found, like you see here, this is the wild type region, and this is double mutant, and this is sample mutant and sample mutant for each site. And we found that the double mutation induce total loss of fixation. So we tried in vivo to analyze the effect of this double mutation in vivo, and what we found that this mutation induce the loss of the activity of the leg antennal element. So we confirmed that this region is important for, it's a region very important for the binding of the activator. Subsequently, we have analyzed the expression of another potential activator, which is rotund. We know that rotund is expressed proximally in the antenna. So we tried to verify first that that rotund is also needed for the leg antenna element. So we generated many clone and we, as you see here, in the region where BAP2 where rotund is expressed, if we generate a clone for rotund minus, we lose the expression of BAP2 and also the activity of our transgene. Subsequently, we have analyzed the sequence of this region that we found that previously that is important for distal proximal expression of our transgene. And we found a potential site for rotund. We verified that this site is able to fix the protein rotund, and if we mutate this site, we lose also the binding, which confirms that this is a very strong site for binding of rotund. We have tested in vivo the effect of deletion of this region, and what we found that the deletion induced the loss of the proximal ring, which confirmed that rotund is important for proximal expression of BAP gene. We have also analyzed the effect of expressing rotund in the distal domain in presence and in absence of this regulatory region. And what we found that when we use one element with the rotund site, we have expression in all the domain distalless. However, if we delete only this this sequence, we lose the expression in the discs. Where, however, this expression of BAP2 is still maintained. This experiment confirmed that here there is a rotund site, and that rotund is a direct activator of this element. Finally, to confirm the role of distalless and rotund in activation of BAP, we have generated clones that express distalless and rotund in the halter. And as you see here, normally the halter don't express the element, the leg antenna element is not expressed in halter. And even if we induce the expression of distalless or rotund alone, there is no expression. However, if we express in the same time in the clone distalless and rotund, the LAE start to be active, and we, as you see here, there is a RFP, which confirms that for the activity of this element, we need the presence of the two proteins in the same time. And this is the model that we have proposed for the regulation of BAP2 gene. We have also analyzed some other sequences that are implicated in repression, and we found that these sequences are important for repression. As you see here, if we mutate all this this consensus sequence for BOL, we have the repression of interring and proximal. To confirm also that BOL is a direct regulator, we created a mitotic clone in which we lose the expression of BOL, and as you see here, when we lose the expression of BOL, there is a derepression of BAB, and in particular for the leg antenna element. And in the same way, if we express GFP, if we express ectopic BOL, it leads to the loss of expression of BOL and also the loss of activity of our cis regulatory element. Recently in the lab, the group of Henri-Marc Bourbon, they have tested the effect of deletion of this element using CRISPR-Cas9 technology, and they have found that this element is more important for BAP2 than for BAP1. However, the flies are still have ectopic sex cam teeth on the tarsal 2, which are typical of BAP2 hypomorphic alleles. Voilà. Thank you for your attention.
Excellent. Cool. Thank you. So Aissette, questions. Miriam Deniki Saladin said she really enjoyed it. Actually, she said it was fun to watch. In the spirit of translational research, is there an example of an instance or a disease condition where CRM manipulation can find relevance?
I have not heard the question. Can you please repeat?
So the CRM and these shadow enhancers, do you have examples where they are important in human disease, for example?
In this moment, I don't remember exactly, but of course, if there is a mutation in a cis regulatory model, it can induce pathology. But I don't know if there is some mutation that is described for a cis regulatory model. I have not heard about that, but probably yes. Normally, yes. Because generally, enhancer, there is not only a cis regulatory element, there is not only one cis regulatory element. Sometimes there is many cis regulatory elements, and there is interaction between this element. So probably one punctual mutation, it's not enough to generate a phenotype, like a stop mutation in coding region or something like that. However, if there is a deletion, sure, we will have a big deletion, we will have a phenotype. And generally, if you have a big deletion, we have also the gene, some genes that are deleted. So it's not easy to relate cis regulatory model mutation with the phenotype. I think it's not so easy. But if in addition, these cis regulatory models are not all known for the moment. there is a few cis-regulatory models that are identified. So first we have to identify this region, and second, we have to understand how it works, with which factor it interact, and then we can start to relate a disease to a punctual or a small deletion or something like that. But first we have to finish to identify all the CRM, and then probably we can start to relate the disease with the cis-regulatory model.
Yeah, no, I agree. It's going to be, I think the idea, as you say, is that probably shadow elements are important in disease, but people like you are actually going to have to figure out what they are first, so we know they're mutated. We know there's a lot of mutations between genes that are important. We just don't know what to do with them. Exactly, I completely agree. So Miriam also asked, since BAP2 gene is conserved, which it is, have you tried severing one of the fly's limbs to see if it will regenerate in place of organ grafting or organ regeneration? Since BAP2 is part of a pathway that's important in regeneration, what about limb regeneration in flies? And what about using shadow elements as part of that?
A second time I have a problem to hear. To hear, I don't know.
No worries, no worries. So, Marianne, would you like to ask this question?
All right, shall I go ahead? I think
Yes.
I'll just ask the question in French.
Yeah. Go
okay, Dr. Baanannou, this was a nice presentation. I enjoyed looking at the appendages regenerating. The clarification I want here is, let's say, for instance, you know now that BAP2 is responsible for limb regeneration. I don't know how you came.
It's not regeneration. It's limb junction formation of junction.
Okay, formation of junction, right? Yeah. Okay, let's leave it at junction formation. So let's say, for instance, you pick a fly and then destroy one of the limbs. Did you do that? And did you see the junctions regenerating? Or did you see the junctions forming back? I don't know. How else to put the question.
The objective of our study was not to test the effect of a cis-regulatory model in generation of a leg. It was just to identify the cis-regulatory model. After, if someone have tested the regeneration of the leg of Drosophila, I have no big idea about this. I think if someone cut the leg, it will not regenerate. This is what I think.
Okay.
It's not probably a brain. There is experimental regeneration for the brain, for some organ, but not for the leg.
Okay, maybe you should try it in your lab, so that we can have a promising future for people with amputated legs.
Yeah.
You know, something like that. I don't know. Thank you for presenting.
Thank you, Marianne. And may I say, as an old-time fly guy, there are old classic, definitely Aissette before your time. There are old classic experiments where you cut the developing disc and it regenerates. There's been work on that, but that hasn't been done in a long time. So Marianne, actually, your ask is definitely possible, but you're going to have to encourage Aissette to take up an old baton and reignite it, or maybe you can do it yourself. So John Oyem says, nice talk. Can you talk about the composition of your rescue assay? Basically, is that how did you rescue BAB1 and BAB2?
Okay. So how we rescue? So we have used the back. It's a big plasmid which contain all the coding region for BAP2, in addition to the intergenic region which contain the leg antenna element. So we have introduced this construction in the context of BAB minus. It means there is deficiency that delete all the two genes in the same time. And we have used the homozygote flies that are deleted for this locus. And in the same time, we have integrated this BAC, which contain BAP2 with LAE, by transgenesis, with using ATTB system. So this BAC will work if it works, and he worked, and will save will allow the expression of BAP2 in the imaginal disc, and then will allow the formation of normal tarsus in the leg and antenna also. So this was the first step, is to verify that the BAC is working. Then we have generate a second BAC by experiment by I forgot recombinating, and we have deleted this region and cis regulatory model, and then we injected this modified BAC, integrate this modified BAC in the Drosophila. So we have two models, one with the leg antenna element and one without this element. So when we have deleted this region, we have lost the rescue that we have we obtained with this BAC. So we confirm that this leg antenna element is necessary for the activity of BAP2 gene, and it's necessary.
Yes, and we have time for one or two more questions. I'm going to ask one by China Orish. Great presentation. Could you explain the GAL4 UAS system? Mm-hmm.
Yeah, okay. So GAL4 UAS, when we will want to drive the expression of one gene in a particular region of the disc, imaginal disc. So we will use a driver. Which driver? When a driver is a gene that will induce the expression of GAL4. So this, and then we introduce a second UAS, and with our gene that we want to express. For example, in our case, we want to express rotund. What we do, normally we found these lines available in the Bloomington or something like that, and we order these flies, and then we recombine what we want to do. Distales, for example, we want to express rotund in the distales domain. We cross distales UAS GAL4 lines with UAS rotund lines. We can change UAS with another thing, and we can change the driver, use another driver. So each time it's the GAL4 will be expressed in the same domain as the driver. That is the principle of the experiment. And then well.
Yes.
I hope it was clear.
Well, so I think, yeah, well, explaining UAS GAL4 in the question session might be a little challenging, so I thought I'd let you take a crack at it. We can, you can definitely do a full job on that in Slack, okay? Because this is really one of the most important tools that we have, right, in this expression. But I am going to thank you. We're going to finish your talk and stay on time. Yes. If people would like to applaud for Aissette, I would certainly encourage that.